you know what your presentation
No.
is?
Oh, is it? Okay, thank you. Okay, all right, well thank you very much to Sydney Vital, Alexander, Susie and Rob Baxter for supporting this work. So I'm working on the Flagship 1 program with Sydney Vital, and my project has been focused on co-targeting insulin-like growth factor receptor and sphingosine kinase in breast cancer. So Rob and myself have just recently published a fairly comprehensive review investigating and detailing the preclinical and clinical studies that have been undertaken to target IGF-1 signaling in breast cancer. And IGF-1R is well established in the literature to be oncogenic in many different types of cancer, and specifically a number of different monotherapies have been designed to target IGF-1R, but the issue with these clinical trials was disappointing in a number of aspects because they just weren't effective in leading to any improvement in disease outcomes. So in order to properly try and work out how to target IGF-1R signaling, it's now becoming evident that there are many other pathways involved that need to be co-targeted in conjunction with IGF-1R. So IGF-1R can be targeted using IGF-1R monoclonal antibodies which target the receptor itself, monoclonal antibodies which target the ligand, and tyrosine kinase inhibitors which actually inhibit the heterodimerization of IGF-1R with the insulin receptor. Now IGF-1R is known to activate many downstream pathways in the cell, and these filter through to PI3 kinase, FAK and MAP kinase pathways, and as you can see on this diagram there are many different inhibitors that can target these IGF-1R signaling pathways. Another pathway which we've been focusing on is the sphingosine kinase pathway, and this is known to be regulated via IGF-1R, and this occurs via activation of phosphorylated activation of sphingosine kinase via MAP kinase. And there are sphingosine kinase inhibitors that have been used, but they are not clinically used for breast cancer. So there are a number of different clinical trials which are underway in the States to try to work out how to effectively target IGF-1R, and these are combinatorial therapies which can target IGF-1R itself or downstream activation, such as using the AKT inhibitor. And these are just a list of some of the most recent trials, and an interesting trial here is using a bispecific monoclonal antibody which can target HER3 and IGF-1R in conjunction. So my study has been aimed at looking co-targeting IGF-1R sphingosine kinase and whether or not this is more effective than targeting those pathways alone. And we've used specific targeted therapies, OSI 906, which targets IGF-1R and sphingosine kinase, targeting using SKI. And we can see that when we do co-target these pathways, there is an effective significant reduction in colony formation over the use of the individual therapies in both ER negative and ER positive breast cancer cell lines. The second part of my project was to try and identify the prognostic significance of IGF-1R and sphingosine kinase in a co-expression setting in breast cancer. So we were able to obtain 236 breast cancer samples from the Australian Breast Cancer Tissue Bank. And these samples were all stains for IGF-1R, phosphorylated IGF-1R, sphingosine kinase and Ki-67. All the samples were manually scored from weak to strong, and Ki-67 with a cutoff of greater than 10% or less than 10% to stratify into low and high cohorts. And when we undertook our Kaplan-Meier analysis, we were able to find that high expression of IGF-1R is actually associated with better disease outcome in breast cancer patients. And this has been reported in the literature. So we were fairly confident of our data, and we were also able to show that when we looked at high co-expression of the two genes, that again we could see a better outcome, disease outcome in women with breast cancer. We were able to show that this is specifically related to ER-positive breast cancers in terms of a high Ki67 expression was also associated with better disease outcomes in women with breast cancer. And when we looked at co-high IGF-1R and Ki67, again we saw the same effect. We went through to sort of try to look at some correlations between these factors, and we could see that IGF-1R and Ki67 are potentially correlated. We were able to show that IGF-1R is also correlated with sphingosine kinase and Ki67 specifically in ER-positive disease, and this was not evident in ER-negative disease, which gives a good insight into the correlationship between the two genes. And we're also able to show that sphingosine kinase is potentially also correlated with Ki67. So lastly, the Flagship 1 program, this has enabled for the facilitation of better patient outcomes by looking at the clinical need for effective combination therapies. This study has allowed for better prognostic significance of IGF-1R signalling and identification of a new IGF-1R correlated pathway. Thank you.
We've got time for a question. Any questions?
No, I'm free to go.
You're free to go. You're free to sit in your Thanks, Alex.
Thanks.
So the next one to introduce is Dr. Amy Hayes. Hayes, she's working with Dale Bailey and our neuroendocrine flagship. Amy? Where's your talk?
Okay, so today I'm talking about the management of neuroendocrine tumours and the clinical and prognostic utility of dual PET. So this is a 38-year-old lady who underwent a distal pancreatectomy in 2011 for a low-grade pancreatic neuroendocrine tumour with a Ki67 of 1%. Within 12 months, her disease unfortunately recurred with liver and bone metastases. You can see some of the liver metastases and bony metastases on the 68 gallium dotatate PET up the top, but on the FDG PET down below, it shows largely non-FDG avid disease, apart from uptake in maybe a couple of liver metastases. So suggesting that the bulk of her disease remains well differentiated. Because her disease begins to slowly progress, she goes on to have 4 cycles of lutate therapy and achieves disease stabilisation with a small amount of tumour shrinkage. Her FDG PET, however, remains relatively quiescent. However, after 6 months, a lesion in the left lobe of the liver begins to rapidly progress, and you can see that it is now not only dotatate avid, but intensely FDG avid. Suspecting the development of much more aggressive disease now, this lesion is resected, and histology returns consistent with a neuroendocrine tumour, but now with a Ki67 of 30%, consistent with grade 3 disease. So she's moved from a grade 1 disease in 2011 to grade 3 disease over the course of 3 years. She moves on to have targeted therapy with everolimus, which has been demonstrated to lengthen progression-free survival in the pancreatic NET population. However, her disease significantly progresses over 10 months, and now she has widespread disease in liver, bone and nodes, and now also with significantly more FDG avidity, which is more consistent with her development of higher grade disease. Fortunately, all of her FDG avid disease also takes up dotatate, indicating that her disease continues to express somatostatin receptors. And she goes on to have 2 further cycles of lutate therapy and has an impressive response. So while well-differentiated NETs have historically been considered quite indolent tumours, we now know that their behaviour can vary quite significantly, and histological grade of the tumours between patients and even within the same patient can be very heterogeneous. Hence, it is now well recognised that predictors of treatment response and prognosis is an unmet clinical need in this population, and histology alone from a single site biopsy cannot be considered the gold standard. Whilst there is a growing body of research investigating the utility of dotatate PET and FDG PET individually in this population, there are very few centres around the world, apart from here in Australia, that are coupling the dotatate PET with an FDG PET. And we think that both scans interpreted together are invaluable. We know that avidity on 68 gallium dotatate PET reflects well-differentiated disease and the relative presence of somatostatin cell surface expression. And these tumours likely behave more indolently, in contrast to FDG positive tumours, which are typically poorly differentiated and carry a poorer prognosis. When the PETs are interpreted together, they are very useful because they can evaluate the entire disease burden, and performing a dual PET has been likened to performing a virtual biopsy. They can assess tumour heterogeneity. They can guide the most informative site for biopsy, for example, the site of most aggressive disease. And by assessing somatostatin receptor expression, they can guide treatment decisions like suitability for peptide receptor radionuclide therapy or lutate therapy. So they can act as a predictive biomarker for lutate therapy. In order to take this concept further and to summarise information from both scans into a single score, David Chan, Dale Bailey and the Flagship 2 developed a novel grading scheme called the NetPET grade. Now, I don't expect you to be able to see the details of this slide, but I do hope it demonstrates the complexity of having to overlay two PET scans simultaneously and interpreting varying patterns of dotatate and FDG avidity. And this is what our current referrers are having to do, and we're determined to make this process more straightforward for them. So the crux of the flowchart revolves around the amount of concordance or discordance between dotatate avid and FDG avid lesions in a single patient. A P1 grade, for example, defines dotatate avid disease that has no FDG avidity, so more indolent, better prognostic disease. In contrast, a P5 grade describes disease that is FDG avid with no dotatate avidity, so poorly differentiated, more aggressive disease not suitable for lutate. And then all the grades in between from P2 to P4 describes disease with varying amounts of dotatate avidity and FDG avidity. So we went on to explore whether the NetPET grade could predict overall survival. We performed a retrospective study looking at 62 of our histologically proven NETs patients with metastatic disease who'd had a dotatate and FDG PET within 31 days of each other. The majority of patients had well-differentiated pancreatic or midgut NETs. As you can see, when they were stratified into three groups based on NetPET grade, so P1 versus P2 to 4 versus P5, there was wide separation of the curves and a significant correlation with overall survival. Median overall survival in the P5 group, which is the FDG avid dotatate negative group, was 11 months. And you can see that median overall survival was not reached in the other two arms, but at 32 months, 75% of the P2 to P4 patients and 90% of the P1 patients were still alive. So in summary, we propose that the NetPET grading scheme will not only simplify complex information and standardise reporting, but we hypothesise that it will help guide management decisions by predicting responses to treatment and allowing for better prognostication at patient diagnosis. Ultimately, we believe the use of dual PET will help to individualise treatment decisions and treatment recommendations in this patient population, which is very much needed by the clinician. Thank you.
Thank you very much.
Thank you.
One question.
You demonstrated the Kaplan-Meier curve.
Sure.
Was that an independent cohort from the cohort on which the NetPET grading system was developed?
Sorry.
So you developed the NetPET grading system?
Sure. Yep.
And then you presented a Kaplan-Meier survival curve on a cohort?
Yes.
Was that a different cohort to the cohort on which the NetPET grading system was developed?
Well, the NetPEC grading system was developed based on our entire population of NETS patients. The concept, we took all the patients that had FDG PETs and 68-gallon Dotec PETs at Royal North Shore Hospital, and we only included those patients that had those 2 PETs within 31 days of each other to make up our cohort for that particular retrospective analysis.
So Anthony, we came up with the classifications on paper and we applied them to the patients.
Yeah.
I think Anthony's right. All clinical scores, you should have internal validation and preferably external validation from a different group, and that's what you're working towards, aren't you?
Yeah.
Good. Thank you.
Thank you.
And next speaker is Crystal Senior from Mark Malloy's group at Macquarie. So thank you very much for presenting.
So just in the folder. Cool. So just hello everyone. I'm Crystal. Oh, sorry. Yeah, Mark does apologise for not being able to be here. So I'll be reporting on the seed funding and what was done with that money today. So our project really worked on the control of the PI3 kinase pathway in BRAF colon cancer, and we did this by using a combination of different inhibitors. So BRAF colon cancer, there are about 15,000 cases of colorectal cancer in Australia per year, and about 10% of these cases harbour a BRAF mutation. Unfortunately, BRAF inhibitors alone are actually not effective for these cases, despite the fact that in melanoma, BRAF inhibitors alone are actually effective enough. Okay, so BRAF colon cancers often show other mutations, so other genetic abnormalities in the PI3 kinase pathways, such as PTEN loss, which is a known tumour suppressor, or PIK3CA activating mutations, and PIK3CA is a gene in the PI3 kinase pathway. So obviously the PI3 kinase and AKT pathways are quite important in these cases because the activation seems to be central for their acquiring resistance to the treatment. So why is this important? Currently, for those 10% of cases of cancers that harbour the BRAF mutation, there is no effective targeted treatment for these people, unfortunately. So previous work from the Malloy group had shown that inhibition using casein kinase 2 worked synergistically with BRAF inhibitors, okay, in controlling AKT activation. However, I do have to note that this work was done in melanoma and thyroid cancers, cancer cell lines. So using that data, we came up with a hypothesis that perhaps addition of the casein kinase inhibitors along with PI3 kinase inhibitors would actually be more effective in controlling proliferation in the BRAF mutant cancers rather than just the BRAF inhibitors or the PI3 kinase inhibitors alone. So the approach that we used was a series of cell lines with known mutations, both just single mutants or double mutants, and upon exposing the cells to drugs and a combination of inhibitors for 72 hours, we determined cell viability. The second thing that we did was mapped the signalling and followed changes in protein phosphorylation by using phosphoproteomics and mass spectrometry to look at those as well. So essentially what was found was that the GDC0941 alone, which is a PI3 kinase inhibitor, was highly effective for all the cell lines, which included KRAS mutants, BRAF mutants, and double mutants, which were BRAF mutant and also PI3K mutant. What was found was that the BRAF inhibitors, along with the CK2 inhibitors, so the BRAF inhibitor we used was dabrafenib, CK2 inhibitor we used was CX4945, was actually more effective than the single agent alone by up to 1.5-fold. So the small amount of data that I'm going to show you, I'm only presenting 2 cell lines, the H229s and the LIM2405s. The reason why we've chosen to show you those is because they're actually the double mutants, so the ones that are probably harder to treat. They both are BRAF mutant, but the H229 is PIK3CA mutant, and the LIM2405 is PTEN null. So we got up to a 1.5-fold better result than just the GDC alone, which is over here. Sorry, you can't really see that up there, but over here we've got the dabrafenib, and the grey is the H229 and orange is the limb. The CX on its own, so the CK2 inhibitor alone, the PI3Ks inhibitor alone, and then we've got the triple combination. Obviously we've done a number of combinations, but I'm just trying to show what's most relevant. So from that, I mentioned that we did the phosphoproteomics as well, and what was found was that there was an upregulation of SRC family kinases, which were activated in response to the PI3 kinase inhibition. So what we then thought of was, in addition to testing these other inhibitors, we would use PP2, which is a SRC family kinase inhibitor, along with the PI3 kinase inhibitor, and we found up to a 2-fold increase. So it worked better than, again, the GDC alone. So what's novel about this is that this combination has not yet been used, in particular in colon cancer cells, and especially for the ones that harbour the BRAF/PI3 kinase mutations. So we found that although the GDC alone was effective, the blocking of the CFKs actually increased the effectiveness. And I will also make a note that the phosphoproteomics did identify other leads yet to be followed up, and one of those was the upregulation of the SFKs. So translational goals, building Sydney vital capacity for colon cancer research, and providing preliminary data for further grant applications, and future directions do include testing in vivo mouse models, and enabling further collaboration with our researchers at the Colling Institute.
No questions from the floor.
There are some drugs that block both sort of upstream and downstream in the PI3 kinase. Do you think about using those, or do you think that you run into toxicity problems because you're already combining something?
Exactly. So in terms of the combination, I know previous work had been done, for example, with the Lie drug. I'm not sure of the number on the end of that. But what we favoured, I guess, because of the toxicity and I want to say the molarity of the drugs that we were using was the GDC. Although I think there was perhaps one other paper in another cancer that used a separate, or different PI3 kinase inhibitor plus a SRC kinase inhibitor as well. But at the moment we are favouring the GDC, and favouring the GDC plus PP2 over the CK2 inhibitors. Yeah.
Is the incidence of colon cancer rising? And if so, is it anything to do with the nutrition diet?
Well, I mean, I want to say yes, absolutely. And the cases are rising for both men and women. And I think the statistic that I had of 15,000 was perhaps maybe even a 2014 statistic. But I do know that they are rising, but I just don't know by how much. But I would probably put down something like diet, yeah.
Yep. Thank you very much.
Thank you.
So move for Haryana Dillon is working in Section 3 with Van Boyle to the presentation.
Thank you very much. Okay, so I'm feeling like the odd one out here. Talking about the implementation of a talking book in radiation therapy departments. The project that was funded is it feasible and can it help? So this was first question is, why are we doing this? So it might surprise some of you to know that low health literacy is very common in the Australian population. About 60% of people in our population will lack basic health literacy skills. Radiation therapy, as you will know, is a commonly used cancer treatment for people with different types of cancers, but many people have difficulty understanding the concepts of radiation and why it's used for different types of cancers. And they also have a limited understanding of radiation therapy, with many misconceptions perpetuated in the community. So, and we also know from work done elsewhere in Australia that information needs a highest at treatment planning and before people start treatment. So, but there are also few interventions in radiation oncology for low health literacy populations. So what we did with our group, with some work that was done at Prince of Wales Hospital and St George was to develop a radiation therapy talking book. So the written document was designed on low literacy principles, so you can see there that it's a lot of white space, easy to use and tabulated so people could work their way around it. This printed resource is supported by an audio recording of the content of the book, so the talking component of this. The reason we went with the talking book component is that there's evidence to suggest that, or to support the idea that people learn better if they both see and hear information delivered to them. And we thought this was potentially something that had a lot of usefulness to different approaches in the community. So we did a pilot study, a pre-post test questionnaire that was done last year and analysed this year. We had 40 patients with a reasonable age range, mostly female, and surprisingly high level of education. But they had and high health literacy. What that study showed was very encouraging for the use of this book in that it demonstrated a significant improvement or significant reduction in anxiety before patients started their radiation treatment, reduced concerns about radiation treatment itself, and also increased knowledge about radiation. So we thought about what we should do with this, and we had considered whether or not we should try and evaluate this in a randomised controlled trial as to some way of improving patient outcomes, but decided it's a pretty low-risk intervention. There's lots of evidence to suggest that improving communication improves and helps with patient outcomes, so there probably wasn't a lot of point just trying to do that. So what we decided to do was move to an implementation project. So our global aim here is to implement the Radiation Talking Book using a particular framework for implementation science as a holistic patient education tool within radiation therapy departments across the Sydney Vital network. And the Paris framework is one which uses the Promoting Action on Research Implementation in Health Services, where we look at the evidence, the context in which the evidence is going to be applied, and what the facilitators are for that. So this meets the Sydney Vital goals by facilitating generation of practice improving research, so delivering and helping patients to get to their radiation therapy much more effectively, which will ultimately improve their outcomes. It facilitates the evidence into practice by helping people do that, as I've just said, and it's building our capacity to do more implementation science and to in fact do more science across the board in whole of department centres. In order to do that. So in the implementation project there are several stages that we're working through with the hospital involved, and the first was to document the current communication and patient education processes in the radiotherapy department and to pilot some potential outcome measures. Accessing or assessing their communication processes both pre and post implementation, then tailoring how the talking book will be implemented in the process for each site. It needs, if it's going to be successfully used, it needs to be tailored to the specific needs of each hospital and the way that they function and the people who work within that. And then we wanted to determine the impact of the talking book on patients in radiation therapy services. At the moment we're working through the process with Gosford Hospital, aiming to implement this as this talking book as a department-wide communication resource in radiation therapy. You may or may not know that Gosford has only fairly recently had a radiation service, and one of the things that they were most interested in doing with us when we showed them our resource was actually outreach into the community. So they're going to use this resource as part of their tools in which they can educate the community about what radiation therapy is and how it's used and delivered. We're also approaching Nepean Hospital about implementation more to try and access a higher number of lower literacy and lower health literacy patients in that process. Westmead Hospital had approached us independently about the potential for using the Radiation Talking Book in and translating it into other languages. And by chance I had contacted or been connected with a clinical psychologist from India who speaks multiple languages, and we're working with her to translate some of the talking book into a couple of languages and pilot that at Westmead as well as a proof of concept that it can be done. In addition, the Targeting Cancer initiative, which you may be aware of, has been set up to develop and disseminate novel education support tools for incorporation into their suite of resources.
about radiation therapy to educate the community. What we're doing with them is that they're incorporating, or planning to incorporate our radiation talking book into their suite of projects. We're also working with the Beyond 5 group who've developed information for head and neck cancer to disseminate this, this project through them, and actively working on funding sources for adapting the talking book for Indigenous Australians in collaboration with Professor Gail Garvey, who is an Indigenous health researcher in cancer, and again have approached internationally looking at doing Chinese translation with Wendy Lam from Hong Kong to see whether or not this particular resource can then be used both locally in our own Chinese-speaking population and in Hong Kong in a group of people from overseas. Thank you. That's where we are.
Are there any questions? Yes.
Is it sensible to use it without the audio as well, just for the
Yes, it is, and that's one of the things that we do with the implementation process, is to train people and work with the staff in particular departments and how they would use this in their communication with the patients. And what we've done is created some sections of the talking book in A3 pages for them so that they can actually then refer to this and use it much more effectively as part of the usual communication tasks that talks that they're doing. But it refers back to the book, so it reinforces to the patients that the information is in the book that they take home with them.
All right.
Does it matter if the narrator is male or female?
Really good question, and to be honest, probably not very much. I think we probably get more variation, you know, it's very individual, and I think overall it doesn't matter that much. But it would be an interesting question to explore. We did sample when we were creating the audio, both men and women narrators, and ended up going with a male narrator.
It's interesting that both Siri and Google use female narrators.
Yeah.
Do you use them? Do you use it?
Maps, Google Maps.
Google Maps, yes, fine.
Thank
Thank you very much, Karen. So next up is Ben Harris, who's working with Kelly Charles at Central University of Sydney. Third year now in your PhD PhD project.
Okay, so my Sydney Vital project is about understanding more about the mechanisms about chemoresistance to platinum drugs as mediated by systemic inflammation, which forms a part of my broader PhD looking into the role of systemic inflammation in predicting patient outcomes in lung cancer patients specifically. So I want to start by highlighting all the people that have been involved in this project at various stages. So it initially began as a highly collaborative project from an NHM MRC grant a few years ago, and now I'm based at the University of Sydney in the discipline of pharmacology under the supervision of Dr. Kelly Charles using some of the funding from this grant, and we closely collaborate with Professor Andrew McLaughlin from the Faculty of Pharmacy. We also engage with Dr. Peter Galetes from the University of Newcastle and several clinicians from the Northern and Concord Clinical Schools of the University of Sydney have been involved, including Professor Stephen Clark and Professor Philip Bill, as well as Dr. Via Fan, and some amazing scientists, including Dr. Vijay Pereira and Annalise Syke, have contributed to this research. So obviously there's a lot of connections between scientists and clinicians, which is essential for successful translational science. And owing to this, subsequently from the initial grant, we've had successful funding from the Cancer Institute of New South Wales, as well as more recently from Sydney Vital. So my PhD mainly looks at lung cancer patients. As you're all probably aware, lung cancer is a huge disease burden in Australia, with about 12,000 Australians being diagnosed predicted this year, as well as 9,000 cancer-related deaths. Even though there have been years of research into improving patient outcomes, there's still a very low survival, 5-year survival of 15%. So clearly we can do better. One of the major drugs given to cancer patients is platinum-based chemotherapy, generally carboplatin. And the problem with these drugs is that, as with many cancer drugs, is they have very narrow therapeutic windows and they are highly toxic. So dosing patients properly is essential. There is some dose individualization efforts. However, these are limited to basic patient characteristics such as age and sex. And even with these guidelines, there are still highly unpredictable and extremely variable patient chemotherapy responses to these drugs. So obviously these drugs need to be further optimized, and scientists and clinicians need to work together to better understand some of the factors that regulate how platinum drugs actually work. And so there is some limited available to us, and hopefully my research can fill in some of the gaps. So through my research, we've been looking at a marker of systemic inflammation called the neutrophil to lymphocyte ratio, or NLR, and have seen that this marker predicts drug behaviour and patient outcomes in non-small lung cancer patients. So in a lung cancer cohort of patients, those patients that have an NLR above 5, indicating they have a high systemic inflammatory burden, seen in the red sort of bars, have an increased carboplatin area under the curve. So this means that they have an increased carboplatin drug exposure systemically. These high NLR patients also have a reduced response to their chemotherapy and a massively reduced overall survival time, with a median survival time about 1 year less compared to patients that have a more normal inflammatory status. And through the research, it seems that those patients with the high NLR with the increased systemic drug exposure also have increased risk of dose-limiting toxicities, leading to the early cessation of their chemotherapy, and perhaps explaining these poor outcomes because they're simply not finishing their chemotherapy treatment. So the Sydney Vital project was looking more mechanistically in vitro to determine how inflammation may be regulating drug behaviour. So the few graphs I'll show you are just for a lung cancer cell line, A549, and one of the things that I was looking at was the ability for these cells to, how they accumulated platinum in the exposure to inflammatory agents. So in this graph, the cancer cells exposed to the pro-inflammatory cytokine interleukin-6 had a reduced accumulation of the cisplatin, one of the platinum drugs. What was also interesting, at the same time that we also co-administered in some of the cells the drug ruxolitinib. So ruxolitinib blocks IL-6 signaling and is essentially, I guess, anti-inflammatory in this scenario, and it was able to restore back towards controlled conditions the platinum accumulation in these cells. I was also looking at how this may be occurring, so looking at the expression of different drug transporters on the membrane of cancer cells that dictate the platinum distribution within the cell. And there were some subtle changes. Some transporters didn't change, others increased. So presumably if they're an efflux transporter, perhaps they are increasing the clearance of these drugs from the systemic, from the cancer cell. But at the same time, there was decreased, other transporters had decreased expression, so perhaps countering it. But obviously the global changes in these transporter expression need to be further determined to determine the net effect of platinum distribution. But again, when we co-administered a blocker of IL-6, we were able to restore towards baseline the original phenotype. And sort of lastly, one of the things I looked at was cytotoxicity. And regardless of the conditions that the cells were exposed to, the platinum efficacy was, there was pretty much no change. So perhaps suggesting that as long as we can get some of the drug into the cells, it'll be efficacious. But if we are having increased clearance and reduced accumulation into the cancer cell, where else is that going? Is it then able to impact healthy surrounding cells, perhaps potentially causing these side effects and explaining the clinical effects that we're seeing? So just to quickly summarize, inflammation is increasingly becoming implicated in cancer outcomes, and some of this research has deepened the mechanistic understanding of how inflammation may be impacting how platinum drugs work. Obviously, further research is needed to complement these findings and to help identify potential drug targets. Perhaps we can be looking at some of these transporters and modulating their function in such a way that the chemotherapy agent is being retained in the cells better and more efficacious, or should we be looking at the immune system and modulating that in some way, perhaps with the use of anti-inflammatory agents prior to or in combination with chemotherapy to restore that normal phenotype. Do we need to do dose modification in these high NLR patients, which is something that we're looking at further based off the initial studies, how much NLR can be predictive of the carboplatin exposure, and how can we use that to dose patients in the first cycle? And obviously, as we continue to work and understand more about chemo resistance and integrate that with new information, we can better manage our patients and have more confidence in the clinical decisions we make about the drugs that we give them and how much of the drugs we get. So obviously all this collaborative research is aimed at trying to improve patient survival and the overall outcomes, and so hopefully in time we'll be able to do that with a better understanding of how chemo resistance is actually working.
Any questions?
There's recently I read a paper that said during the chemotherapy of breast cancer, if the patients exercise, they had a bad outcome. Would you suggest that this might be a mechanism why that outcome was improved with the exercise?
Why it was improved with exercise? If it
would be decreasing the systemic
I don't know if there's a direct relationship, but I assume that'd be generally healthier and maybe that is improving. A lot of the high NLR patients tend to appear sicker anyway and have a worse performance status, and maybe they're not able to do exercise or they need some kind of exercise to reinvigorate their immune system in some way, but I'm not completely sure, sorry.
Rory?
Still just a bit confused. Does the cancer induce a systemic inflammation, or is the systemic inflammation there and makes the cancer worse?
I think it's in terms of colorectal cancer and some cancers, there's definitely an inflammatory component. And I think it's a bit of both, that somehow it's already there and it's propagating the cancer, but as well as patients are reacting differently. So not all stage 4 patients have high inflammation. Some don't have high inflammation and do quite well. So we also think that it's a marker of something else. So perhaps cachexia or some other phenomenon, and that is related to inflammation in another way, and this is just a marker of that kind of behavior. So yeah, I don't know. Yeah.
Watch this, please. Can we just stop you there, because I think we need to go on with the scientific presentations.
Okay.
And the next one is Dr. Jing Zhao from the Melon Cancer, Thyroid Cancer Group. Jing?
Thank you. Firstly, I would like to thank the Sydney Vitals for providing us the seed funding to make it possible for us to initiate this study to look at the associated non-coding RNAs in medullary thyroid cancer. Medullary thyroid cancer is a rare neuroendocrine tumor with poor prognosis. It originates from the very small population of C cells, and currently surgery remains the only curative treatment. Its overall 10-year survival rate for patients with stage 4 disease is around 21%, and this survival rate hasn't been improved over the last 30 years. Gain-of-function mutations of the RET proto-oncogenes has been recognized as the key driver of MTC tumorigenesis. This has previously been targeted with tyrosine kinase inhibitors. However, its therapy efficacy in clinical trials has been very modest. The human genome studies in the last decades have shown that the coding genomes accounts for less than 2% of all genomic genomes, and the non-coding genomes plays an important biological role in cell phenotypes. We are interested in long non-coding RNAs, and long non-coding RNAs are defined as transcripts of greater than 200 nucleotides in length with known protein coding potentials, and the long non-coding RNA genes comprise of around 26% of all human genes. So recent studies have shown long non-coding RNAs contribute each of the 6 hallmarks of cancer phenotypes. They interact with DNA, RNA, and protein to regulate gene expression and to exert cellular effect via great diversities of mechanism. Compared with all other cancer types, non-coding RNA studies in MTC has been hampered. Probably this is due to the unavailability of normal control C cells as a differentially expression comparator. To our knowledge, so far, no publication about the long non-coding RNA studies in MTC has been available. So to this end, we hypothesize that many long non-coding RNAs will be differentially expressed following RET blockade in MTC cells. Furthermore, we propose that specific RET-associated long non-coding RNAs might play fundamental roles in MTC tumorigenesis and modulation of the TKI responses. Manipulation of long non-coding RNA expression might present a window of therapeutic potential. So based on all these hypotheses, our research project includes 3 parts. Firstly, we would like to characterize RET-associated long non-coding RNAs
via RNA sequencing of MTC cells before and after blockade. We plan to inhibit the RET activity using the tyrosine kinase inhibitors and XCONT modified gap-mirrored antisense inhibitors. Second part of the project, we would like to perform gain of function studies or loss of function studies to explore the cellular effects of the candidate functional long non-coding RNAs and identify their molecular interacting partners. Finally, we would like to assess the therapeutic potential of all these identified functional long non-coding RNAs alone or in a combination with tyrosine kinase inhibitors in MTC xenograft models. With the M3, we have confirmed the collaboration with the Ingenic Biotechnology Company in Sydney, which has developed unique targeted nanoparticle delivery vehicles. This technology has been employed in several cancer types, including our studies to deliver chemotherapeutics and microRNAs in adrenocortical cancers. So we are employed this technology in the MTC studies with this project. At the moment, we are in the processing of preparing all the RNA samples from the MTC cells before and after red blockade using tyrosine kinase and antisense inhibitors. Here is the Western showing to confirm the red blockade using the tyrosine kinase inhibitors demonstrating the significant reduction of the phosphorylated ERK in the MET kinase pathway. So following the RNA sequencing, we plan to only to shortlist all those differentially expressed long non-coding RNAs which is common to both the red blockade in the studies. So that's what we've got so far. Thank you.
Any questions? Yep, right there.
This unique one is it derives from bacteria. So after genetic mutation, they kind of like empty all the genetics components of the bacteria, and then they can package either chemotherapy microRNA, and we have tried long non-coding RNA in the different projects. So once it's the unique part of this nanoparticle is attached by a bispecific antibody, which one arm targeting the cancer cell surface receptor, one arm attached to the polysaccharide of the nanoparticle. So in that way, the delivery will be more targeted, and in terms of chemotherapy treatment, you can use less dose of chemotherapeutics.
So you are making this non-coding
with a collaboration with this biotechnology company, Ingenic. Yes.
Thank you very much.
Thank you.
So next up is Janish, third year SVCC, working with Professor David Marsh, Harmonic Cancer Group. Thank you.
Thank you very much. So I'll start my talk with the pie graph, which are sometimes famous in science. So this is the latest annotation of human genome, just published in 2015. And the number of protein coding genes which we have, it's still about 21,000. It has stayed like that since early 90s. But recently, the RNA sequencing and other experiment has shown that the long non-coding RNA, which Jingting talked about, the number of them has actually more than double than the current estimate of protein coding genes. So the long non-coding RNA are transcript which doesn't have much protein coding capacity, and they are longer than 200 base pair. And there are also other interesting genes such as pseudogenes and TUCP, which is transcript of unknown potential, coding potential, which also has a small fragment in fraction of the human genome. So I'll mostly focus on the long non-coding RNA in the talk. So long non-coding RNA have some crucial role in the human biology. For example, the gene here called XIST, X-I-S-T, that has a crucial role in females, that they silence the whole entire X chromosome by epigenetic modification. Other long non-coding RNA such as hot air has a role in in cancer progression as well. Some long non-coding RNA has a role in splicing. An example is MALAT1. Some non-coding RNA can make a triple helix with DNA, and as doing so, they destroy the binding site of a transcription factor and essentially inhibiting the transcription. Some non-coding RNA work as a micro RNA sponge and stop them from working at degrading other messenger RNA. Some of them work on scaffolding and bringing two proteins together. So there are some very fundamental roles already been known, and this is a figure from 2012. And this is the latest figure from 2016, an overview which Jingting talked about as well. Having long non-coding RNA having important role in all 6 hallmark of cancer. Now I want to point your attention to P53, which has a role in apoptosis through long non-coding RNA, as well as a couple of genes in a growth repression. Okay, so our hypothesis was we wanted to look at non-coding RNA, and especially mutant P53 background. So P53 is universally mutated. Almost 100% of the cancer patient, ovarian cancer patient, have that, and obviously it has a big role in this biology of ovarian cancer. And our goal was to understand non-coding RNA coming from the mutation in P53 and hopefully identifying some oncogenic or tumor suppressor long non-coding RNA, as well as understanding their biology. So we decided to do this with a drug called APR246, also known as PRIMA1-MET, and this drug has shown to bind to mutant P53 and reactivate some of the wild-type function. So just very briefly over the mechanism, APR246 goes through the cell membrane without any special pump. It just diffuses through it, and in the cell it gets converted to an active form known as MQ. And this MQ can bind to mutated, unfolded or mutated P53 protein, and it refolds the structure. And as a result of its canonical genes such as P21 and BEX are expressed, and you have a typical apoptosis happening through this refolded P53. On top of it, drugs such as cisplatin, they induce mutant P53, and so the levels of P53 is higher, and as a result, MQ is likely to work more efficiently on the higher level of mutant P53 and inducing apoptosis. So our hypothesis was that if the normal genes such as PUMA, P21, and other genes are expressed by this drug, what about the long non-coding RNA? Can we use this drug to discover non-coding RNA? And that's the hypothesis we are testing. So to do so, we screened a couple of ovarian cancer cell lines for P53 expression, both at protein level and messenger RNA level. And this was actually Christie's data. And we found that 2 cell line Kuramochi and OVCA3 had a higher P53 expression compared to other. And because of the higher expression, we hypothesized that the drug will work better on this cell line. And on top of it, the previous drug, APR246, I mentioned, it also synergizes with lots of other drugs, such as cisplatin. And in ovarian cancer, almost 80% of the patient develop resistance to cisplatin within 2 years. So we wanted to understand the mechanism, how APR246 synergizes with cisplatin and also restores drug resistance in some cases. So we had 2 cell line, again, as I mentioned, OVCA3 and Kuramochi, and we tried a couple of combination of drugs, so APR and cisplatin over time, and the time point optimized at 48 hours. This is the point where we look at synergy. And we found a concentration combination in OVCA3, 70 micromolar APR plus 8 micromolar cisplatin, and in Kuramochi, 88 Shoten plus 20 micromolar cisplatin. So this is our drug combination. And then we did a time course study on those combination in Kuramochi shown in the top and OVCA3 at the bottom. And we found that in a Kuramochi cell line, there is a significant increase in response genes such as P21 and GCLC at 24 hours. And in OVCA3, this response happened a little bit earlier at 12 hours. So we have all these 4 conditions, so vehicle-treated, APR alone, cisplatin alone, and their combination in a 2 cell line and 3 biological replicates, a total of 24 samples. And we just sent those samples at Carbon for RNA-seq and try to understand the mechanism of synergy and how APR246 works. Hopefully we'll have the data by early next year, and fun begins. Thank you very much. I want to acknowledge my supervisor, Debbie and Patsy, as well as Alex, Christy, Sergey, Giles, helped me out a lot with flow cytometry, as well as a collaboration at the Garvan, Nanad, James, and Associate Professor Mark Settinger, as well as Sydney Waddle and APA for sponsoring. Thank you very much.
Congratulations. Any questions for we're eagerly awaiting the results.
Yeah.
Yeah. So this last year, you're going to finish?
Yes.
You're going to finish this.
That's what I said last time, but
what are the barriers? Do you think? Is there, are you on time? Is your timeline okay, or are there any barriers?
Actually, I'm late because of that. The drug APR-246, it has a really funny dose response curve. So at some concentration, it keeps all the cell alive, and if I do double that concentration, everything is dead. So I had to play a lot with the concentration correctly, as well as we found the initial assay we used, something called MTS assay. It wasn't really capturing the cell death we were seeing, so we're doing everything by flow cytometry. And all of this kind of happened with talking to the developer of the drug in Karolinska Institute, I believe it is, and also some people in Peter Mac. So I have definitely had some troubleshooting as well as getting enough RNA for sequencing as well. So.
And the troubleshooting in itself, does that give any answers, or is it just troubleshooting?
Well, look, I'm not a clinician per se, but I assume if you can't predict the dose response curve, right, how is it going to go in the clinic? That's my major concern. And this drug is in phase 2 trial, and they have said up to 200 micromolar. So this is about 3 times more than what I'm using. That's okay. So that's my main concern, and especially we have cisplatin already given to the patient. So I don't know how it will kind of go. I assume you will start with lower and see how toxicity. Okay, cool. Thank you very much.
And the last one before lunch is Samuel Brennan from UTS with Dr. Martinello's group. So thanks very much for presenting.
Thanks. Well, yeah, good afternoon, everybody. I'm Sam, and I work at the UTS Translational Cancer Research Group. And today I'm going to be pitching you the idea of a liquid biopsy for prostate cancer. So I'm sure the first question on your mind is, why would that help? Well, the problem is that about 3,300 Australian men will actually die of prostate cancer this year. And in fact, about 22,000 new cases will get diagnosed. So it's a very high incidence cancer as well. These numbers are high, and they are on the rise as well. I think chiefly because the male population in Australia is aging, and age is the most significant risk factor for developing the disease. But there is also quite poor prostate health check compliance. This involves things like the prostate-specific antigen tests, the fairly infamous digital rectal exam, and also the prostate biopsy. Now, these are all moderately to highly invasive medical procedures, and it is essentially a deterrent for men to getting to the clinic at a stage where their cancer may in fact still be curable. Now, compounding this problem, the PSA test has no prognostic value whatsoever, and it is not always reliable. It can lead to overtreatment where a relatively benign tumour gets a far more aggressive treatment than necessary, and that will lead to a lower quality of life for the patient. And it can also lead to underdiagnosis where a serious cancer can fail to be detected. Now, all of this gives us the conclusion that we need to come up with non-invasive tests with greater sensitivity if we're going to improve early diagnosis and reduce the morbidity and mortality that's caused by prostate cancer each year. And here's the hypothesis that we've chased down to try and achieve that aim. Now, exosomes are released from cultured prostate cells and from circulating exosomes in bodily fluids. These will contain protein, RNA, DNA, all those biological macromolecules, and these signatures can identify different cancers. We hypothesized that microRNA present in the secreted exosomes, I'm going to be calling these exomeres from now on, are in fact reliable biomarkers for the early prostate cancer diagnosis. And you can see on the left, we've actually chased urine in a pretty big way, because urine's in almost constant contact with the prostate, and that would allow the prostate cancer cells to secrete their exosomes directly into this fluid, which we can then non-invasively take from the patients. And here is the methodology we've used to pursue this. So the first thing we did was hypothesis generation using cell culture exosomes, where we grew human prostate cancer and normal prostate cell lines. We performed exosome isolation from them, got the RNA out of the exosomes, and did some fundamental quality control steps. Then we generated our non-coding RNA profile using microarray analysis. Now, once we'd done that, we began hypothesis testing in human samples. So this involved recruiting prostate cancer patients and healthy donors. We got urine from these people, and isolated the exosomes and subsequent RNA from this urine. We were then able to validate our biomarker candidate profile using PCR, and that has allowed us to begin developing a novel, non-invasive qPCR-based diagnostic and hopefully prognostic test. There is a summary of the conclusions that we've made so far after a great many optimizations and profiling steps. The first thing we learned is that a novel cell culture exomer signature, in this case consisting of 12 exomers, was able to distinguish prostate cancer cells from normal cells. However, we found that this actually wasn't very effective when we looked at it in human samples. So it did give us a lot of potential functional information, but not much beyond that. So we redesigned our study using a more powerful technique, and we came up with a novel urine exomer signature, this time consisting of 24 exomers, and this was able to distinguish prostate cancer patients from healthy donors, which is obviously the foundation stone of a diagnostic assay. But the most exciting thing we found was that at least 4 of these urine exomers were prostate cancer specific, and they were significantly downregulated in the patients after they had a radical prostatectomy, which is removal of the cancerous prostate. So this told us that we might be able to develop these 4 markers in particular into a treatment response type of marker where you're a clinician monitoring the progress of your patient by taking urine samples successively during the course of their therapy. If you see these 4 markers go down, you can deduce that it is an effective intervention. Conversely, if you see the levels of these exomers stable or even go higher, you'll know that you need to try something else because it's not working. So where we are right now is designing a custom urine prostate cancer exomer open array type of setup, and we've got 2 retrospective patient cohorts that we're going to push through with that. So that's where we are right now. I'd just like to show you our multidisciplinary team that's helping us with this, and I'm happy to take any questions now. Thank you.
With prostate cancer, it's often not as important whether or not you've got cancer as whether the cancer is aggressive or whether it's something that's appropriate to monitor. Any clues in your signature or exomers about whether you can tell aggressive from indolent cancers?
All right, not yet. We need to get, it's a matter of really validating this with large numbers of patients. Now, what that is much more of a pilot study type thing where we've identified the profile that we're going to try and validate in large numbers of patients. So hopefully it will come out of that. But I can't say yes or no yet with the profile I have.
Any other questions? Good. So this concludes the pre-tea break session. Can I just ask that you all rejoin us at 2:45, and then we'll reconvene and for the last session. Thank you very much, Sam. Just asking, have you finished or?
I'm very, very close. I'm actually I've got the first draft of the thesis, yes.
Good, so that's been the baby and
Yeah.
And then are you going to stay on?
Yeah, I'm staying with Rose for next year. I'm training up a student as well, so I'm trying to do more of this.
That's going to be so next PhD?
Sorry?
Next PhD?
Yeah, so he'll be doing ours next year, the guy that we've got on board right now, but he's very, very after that, so
Good. Good. Thanks very much.
Nice.
There are so many myths that are both seeking to monitor. You sit with a very big you think so invariably so or other evidence to show that yes, the gene gets expressed at such a low level that you can't see by QPC. Right, but by the same token, that's got a sequence 1,000, and every sequence 100 times. These times it's like 4 years. And then it looks like that kind of 15. So that's why it's tricky. I guess the other one is just that in terms of expression, if your mu is phenomenally low and the target set for going off and you've shown that effect, you need to understand physiologically so they're expressing stuff and your mu it's not very effective.
Hi, I get off.
But if it's a transcription factor or slow down,
Did you need to get a presentation on?
No, no, no. Just wondering, is the last presentation not happening, or is it happening after the...
Fred? I'm not sure what happened to Fred.
Yes, because I don't think it's finished the trial, someone said, so yeah, I just wanted to know if it's not happening.
I'm not sure. Someone, April, who's here, who might be from the same unit, might know, but yeah, I'm not sure what happened to him.
So, your biometricians can review what is the chance or the probability of your mirror of interest being a potential false positive. So they can look at the sequence context, they can look at the other genes that are close, the other mirrors that are close to it, look at their expression.
Hi.
And look at your
We had this going before.
We don't want to use it.
Oh, we don't?
No, because we want you to keep talking to the microphone and use that as the mouse.
Okay, I see.
So it's been forgotten.
Okay, verboten. Das ist verboten. Right, minimize eye contact also.
I did a lot of work with isomers. So
Cool.
Cool. I was going to shut down the session.
a lot of those, many of them are real, many of them are shadows of reality.